All right. I think we have come to our last speaker of our today's session. We would like to feature our last speaker, Miss Nurhamimah Misuan from Monash University. She is currently a final year PhD student at Monash University, Malaysia. Her research interests mainly focus on venom cytotoxin, which is another three-finger toxin. And currently, she mixed both computational and wet experimental work to study the interaction between cytotoxin and death receptor. So with her work, she hoped that in future, there will be some kind of alternative drug for pharmaceutical applications to combat the cytotoxic effect during cobra envenomation. So I'll now like to invite Ms. Hamimah to share her presentation.
Right.
Ms. Hamimah, over to you. Yeah.
All right. Thank you, Dr. Michelle. So give me a second to set up everything. All right. Okay. Hello and good morning to everyone in UK and good afternoon to everyone here who came today. So first of all, I would like to thank the organizing committee for inviting me here today. And my name is Nurhamimah Misuan. I'm a third year PhD student from Monash University, Malaysia. And what I would like to talk today is about ensemble-based virtual screening of cobra venom, cytotoxin, and death receptor. So I'll begin my talk by introducing to you my protein of interest, which is cytotoxin. So cytotoxin is a non-enzymatic three-finger toxin present only in cobra, and approximately 40 to 60% of the dry weight of the cobra venom is cytotoxin. So it plays an important pathophysiological effect in cobra envenomation, especially the necrosis. Besides, cytotoxin is a basic amphiphilic protein with 62 amino acid residues in a single polypeptide chain. It is interesting to note that the intramuscular viability of cytotoxin is only 40%, which indicate that it has higher binding affinity towards the site of administration. And lastly, there are two types of cytotoxin, which are P-type and S-type. And P-type is more cytotoxic compared to the S-type. So to continue, the exact mechanism of cytotoxicity of cytotoxin remain inconclusive due to a few conflicting hypotheses. So in this paper, Ibrahim and his fellow researchers, they come with a question whether cobra venom cytotoxin is an apoptotic agent or necrotic agent. So when we are discussing about apoptosis, some studies showed that cytotoxin induce apoptosis towards several cancer cells, such as human breast cancer, MCF7, and MDA-MB231 cells, human leukemia, K562, U937 and HL60 cell, and human hepatocellular carcinoma, HEP2 cell, human lung adenocarcinoma, A549 cells, and also oral squamous cell carcinoma, CA922. So the apoptosis induced by cytotoxin activated certain intracellular signaling pathways such as caspase-mediated apoptosis, JAK2 PI3K pathway, FAST pathway, and the elevation of Bex-BCL2 ratio. In addition, the toxin also induces mitochondrial lysosomal membrane permeabilization and endoplasmic reticulum stress-mediated pathway. So on the contrary, another researcher, Fofanov and Wu et al. Found that cytotoxin may cause direct cell lysis, which may lead to unfavorable cell death similar to necrosis. The necrosis triggered by cytotoxin was shown to cause lysosomal cathepsin B protease leakage. So it is noteworthy that the apoptotic effect of cytotoxin may only be observed in a very limited range of concentration. And the cell death pattern rapidly switches from apoptosis to hypothetical necrosis when the concentration increases, as shown by the paper I show here. So nonetheless, such transition mechanism of cell death are not known, as it is very unclear if the penetration of cytotoxin into the membrane is also a concentration dependent process. On the other hand, a study by Teo and Yap, however, revealed that cytotoxin exhibit concentration-dependent cytotoxicity via caspase-activated mitochondrial mediated apoptosis on MCF cell 7 without transitioning to necrosis. This study by Teo and Yap contradicted the study by Fovanov and Wu above, where they stated that cytotoxin induced necrosis. And another study by Hugh and Yap reported that as the concentration of cytotoxin increases, the cell death transition from apoptosis to necroptosis. So this study supported the previous study by Teo and Yap, where they mentioned that no necrosis occurred when the concentration of cytotoxin increases. Thus, it sparked my interest that cytotoxin might interact with death receptor, which can cause all these apoptosis and necroptosis reaction that have been studied by our fellow researchers. So before we continue further, let me tell you how I got my conserved cytotoxin sequence, and we'll call it CT from now on. So this table showed the conserved CT sequence obtained from multiple sequence alignment. It was found that the sequence identity is the same as the cytotoxin 3A from Naja-Atra. And this conserved CT sequence was then used to determine its three-dimensional structure. So this figure is the 3D structure of CT determined from modeler software. It followed the native structure of another 3-finger toxin family, which include 5 beta-plated sheet with 3 loops. So you can see there are 3 loops and with 3-finger structure here. So this is the Ramachandran plot. This Ramachandran plot showed that the 3D structure of CT constructed before did not deviate from the native structure because 90% of the amino acid residue reside in the favorite region of the plot. So a good model can be defined, for a model to be defined as a good model and closer to the native structure, at least 90% of the residue must be reside inside the favorite region of the plot. So yes, from this plot, we can see that our CT is actually a good model and closer to the native structure of other 3-finger cytotoxin. So the upper region of this plot is the core region of beta strand, while the lower region is the region for alpha helix. So as we can see in this figure, 80% of the amino acid residue reside inside the beta strand core region, which indicate that the cytotoxin is predominantly consisted of beta-plated structure. So this also corresponds to the native structure of cytotoxin, where mostly it consists of beta-plated sheet. So as you can see here, there is serine there is serine 29 here. So this serine 29 show that our cytotoxin is an S-type cytotoxin, and this correspond to the finding by Dubovsky et al., where they found at position 29 there is a serine residue where it conclude the S-type cytotoxin. So in addition to that, physicochemical properties of CT was also determined as shown in this table. So from this table, we can see that CT has 62 amino acid residue with a molecular weight of 7,005.52 grams per mole. Another physical-chemical properties of CT listed in this table is the theoretical PI value, which is 9.38, where it showed that CT is a basic protein, and this was supported by the finding by Kumar et al. The next properties is the instability index, or II, which is 37.71. So this index indicated that CT is a stable in nature, where any value more than 40 will be considered as non-stable. The next properties is aliphatic index, or AI, where it is the relative volume of aliphatic side chain that occupied the protein. The AI for CT is 78.39, which is considerably high for cytotoxin, as mentioned by Panda and Chandra in 2012. So this AI also corresponds to the thermal stability of a protein. The higher the AI, the more thermally stable the protein. Since the AI index of CT is high, so we can say that CT is a thermally stable protein. The next property is the grand average hydrophaticity, or GAV, where it is the sum of all hydrophaticity values of all amino acid divided by the number of residue, where it tells the hydrophobicity nature of the amino acid. The more positive the GAV value, the more hydrophobic the amino acid. So from this table, we can see that the GAV value of CT is negative 0.021, which indicate that CT is a hydrophilic protein in nature. And the last physical-chemical properties of CT that been studied also is the extension coefficient, or EC, where it is used to show how much light can a protein absorb at a certain wavelength, especially 280 nanometer. So the EC can be calculated from the presence of tyrosine, tryptophan, and cysteine residue in a protein, since these amino acids are better at absorbing light at 280. So the EC of CT can be considerably very high, which is 4,970 per molar per centimeter, due to the presence of very high cysteine residue, which is 12.9%, and tyrosine residue, where it has 4.8% from all 62 amino acid residue in CT. So moving on is the secondary structure prediction of CT. This figure showed the predicted secondary structure which are present in CT. So from figure 4, the dominant secondary structure is coil, where 42 residue make up for that secondary structure. So the next dominant secondary structure is beta strand. So there are 5 beta strands present in CT. So the presence of this beta strand resembles the native structure, where it has the presence of beta strand in its core. So the red boxes indicated the amino acid residue in CT, which forming the beta strand. And from all 20 residue which make up the beta strand, 6 of the residue happen to be inside the functional loop of CT. And the residues asparagine 4 and 5, tyrosine 12, methionine 27, and valine 28 and 36. So this prediction also support the 3D structure of CT I showed before. So we'll move to the main point of my talk today, which is the RMSD-based screening of potential death receptor target. There are a few reported death receptors, such as CD95 or FAST receptor, TNFR1, TNFR2, TRAL1 and TRAL2, and also death receptor 6. So this death receptor belong to a protein superfamily called tumor necrosis factor superfamily, receptor. Since they belong to the same family, this death receptor exhibits structural similarity. So to find the best receptor that interacted with CT, we introduce a few selection criteria, such as the number of functional loop of CT involved in the interaction, low Haddock score, Z-score, and RMSD value, and also the characteristic of each death receptor. So based on this criteria here, we concluded that we see that TNF-R1 appeared to be the best dog model with CT compared to the other receptor. So moving on, this table showed the few score obtained after the docking was completed. So I'm going to highlight a few important score needed for data analysis, which were the docking score or the head-up score, the RMSD value, and also the Z-score. So to select the best receptor to interact with cytotoxin, docking score here, the docking score can be used. So we can see here, the R6 appeared to have the highest docking score when docked with cytotoxin, while the lowest docking score was obtained by CD95. So the head-up score is defined as the combination of various energy and buried surface area when the docking takes place. Typically, the lower the docking score, the better the fit between the two docking structures. However, we exclude CD95 and the RC, firstly the RC because the docking score is too high, so we exclude it. And also CD95, even though it has the lowest head-up score, we exclude it because it only has two loops of CT interacted with CD95. For the best activation, we need all three loops to be involved in the interaction between CT and deaf receptor. So another selection criteria to select the best receptor is its RMSD value. Typically, when we compare to superimposed protein structure, the percentage of similarity between two structures are considered to be higher when the RMSD value was below 2 angstrom. The RMSD value for most of the deaf receptor when docked with cytotoxin was, you can see here, it was actually higher than 2 angstrom except for CD95 where it lower than 2. However, it is ambiguous if we only consider the RMSD value to determine the best docking structure. This is because RMSD value has a very few shortcomings. The first one, its dependence on the minimization method and the plan to exclude non-equivalent regions. The second one, the alignment between two structures may differ in different situations. And also the third one, if there is an increase in the molecular weight or the size of the system, RMSD value can lose its sensitivity and bias measurement may occur in order to choose the region which can fit between the two structures. So therefore, we introduce another selection criteria which is the Z-score to help in the selection of the best receptor docked with cytotoxin. So the lower the Z-score, the better the head-up score, since this indicates the standard deviation of the head-up score of a given cluster separated from another cluster. So from this table, we can see that, CD95 also has the lowest Z-score, but as I mentioned earlier, we exclude CD95 from the best receptor dock because it only has two loop interacting with two loop of CT interacting with this receptor. So we can choose CD95 as the best receptor to interact with CT. So this table, table 4, listed the deaf receptor and their characteristic. From this table, the NF-R1 does not possess a deaf domain or DD, which is important in transducing the apoptotic signaling pathway to the cell. Therefore, we exclude the NF-R2 further from investigation. So we also exclude the R6 from further investigation as it was suggested by Gurchadi and Gores in 2009 that DR6 was the less potent deaf receptor compared to the other receptor, even though it has the presence of DD. So in view of the characteristic of the deaf receptor, the involvement of the functional loop, and the summary of the head-up score, the Z-score, and the RMSD value, we see that TNF-R1 appeared to be the best dock model with cytotoxin. So this TNF-R1 and CT will be used to investigate the assemble clustering between TNF-R1 and CT for further screening. So based on the molecular docking selection of potential death receptor, we determined that TNFR1 was the most compatible receptor with CT. So we do the ensemble-based screening of CT and TNFR1. It was performed with molecular dynamic simulation and followed by the RMSD-based clustering analysis before we subjected again to protein-protein docking to determine the best ensemble conformation. The selection criteria were the same as the selection of the best death receptor, except we omitted the characteristic of the death receptor and the number of functional loop involved since TNFR1 was already selected. RMSD-based clustering was performed to select the most relevant representative conformational ensemble of CT and TNFR1 from the MD trajectories. So a total of 4 clusters of CT from this figure, CT1 to CT4, and 5 clusters of TNFR1 from this figure B, TNFR1 to TNFR15 were obtained. So these clusters of CT and TNFR1 were subjected to molecular docking to select the best conformational structure and the best conformational ensemble where they were selected based on the head-up score, the low head-up score, the low RMSD value, and also the low Z-score. So from all these 20 docking possibilities, we listed the best 3 conformational ensemble in the next slide. So this table summarizes the docking parameters of the 3 selected conformational ensemble of CT and TNFR1, which is CE1, CE2, and CE3. So similarly, we rank the conformational ensemble based on the head-up score, the Z-score, and also the RMSD value. So from all the parameters mentioned above, the conformational structure of CE3 fulfill the criteria of the best ensemble conformation, which exhibit the lowest RMSD value and the lowest Z-score, despite it having the highest having the highest head-up score. So, but when we compare between all these 3 conformation, the head-up score can be it was more comparable. So, yep. So that's why we choose the CE3. And in addition to that, the van der Waals and desolvation energy of CE3 conformation was the lowest, which indicated the energy stability of CE3. So aside from the ensemble-based docking between CT and TNFR1, the interaction mapping site of CE3 is done to determine if the interacting site of CT on TNFR1 complement with its ligand, which is TNF-alpha. So this figure showed the amino acid residue on CA3 on the amino acid residue of CE3 on CT, which interacted with TNFR1. So this table showed the amino acid residue on CT which interacted directly with TNFR1. So of the residue of CT showed in this table, approximately 56.1% resided in the functional loop of CT, as it was highlighted in the yellow color here. On the other hand, there were a total of 41 residue of TNFR1 interacted with CT, where 65% of them were identical to the TNF-alpha ligand binding site of TNFR1, and this one is highlighted in the green color from this table. So this finding suggested that there is a possible interaction between the ligand binding site on TNFR1 with the functional loop of cytotoxin. And this interaction can induce the formation of death-inducing signaling complexes, thus activate the caspase signaling cascade. So nonetheless, the activation of apoptotic signaling pathway for TNFR1 was relatively slower compared to the fast receptor, because fast receptor is famously known as the main apoptotic cell death receptor. So since I showed the interaction mapping between CT and TNFR1, the intramolecular interaction between these two proteins also can be determined. So this table showed the type of intermolecular interaction within the binding site of the conformational structure CE3. So both van der Waals and hydrogen bonding were the main contributor to the intermolecular interaction between CT and TNFR1, and this suggests a non-covalent binding of CT to TNFR1 for a reversible reaction, which helped to establish a binding between CT molecule and TNFR1, followed by deactivation. And my next point is the binding affinity between CT and TNFR1. So binding affinity is an important criteria to understand the interaction between two molecules in a system. So it can be defined as the strength of interaction between two molecules in a protein-protein interaction. So this binding affinity was done what this binding affinity prediction was based on the 3D structure of CT, and it was computed using the Prodigy web server. So from this method, we use the CE3 conformation, and this result on this table shown that the delta G, or the binding affinity, is negative 12.2 kilocalorie per mole, which show a relatively lower binding energy. And also the dissociation constant, or KD, is 1.3 to the power of negative 12 molar, which also significantly low, where typically a good protein binding affinity ranging between 10 to the power of negative 6 to 10 to the power of negative 9. So since CT was predicted to have the KD of 1.3 to the power of negative 12 molar, so we can conclude that CT has a high binding affinity towards TNFR1. So in conclusion, the lower the binding energy and the KD, which is the dissociation constant, the better the binding affinity between a protein and its target. And this is the conclusion of my talk today. So CT has 62 amino acid residue with a typical 3-finger toxin structure. So the CT is an S-type cytotoxin, and RMSE screening of potential drug receptor interacted with CT showed that CT keen to interact with TNFR1, and most of the intermolecular forces between CT and TNFR1 are van der Waals and hydrogen bonding. The predicted binding affinity between CT and TNFR1 is negative 12.2 kilocalorie per mole. And from this finding also, we can see that CT can possibly activate TNFR1 cell death signaling pathway. So, yep, this is the references that I use. And yep, thank you for listening to my talk. And yeah, I open the floor to question.
Okay, thanks, Hamima, for presentation. I've seen 2 questions coming in. The first question is regarding aliphatic index. Does a value of 78.39 mean that the CT protein is theoretically stable up to 78.39 degrees?
I'm not sure about this one, but maybe I need to figure out more on this aliphatic index also, because it stated that yeah, it's a stable protein in nature in heat also. So yeah, maybe I need to find more about this aliphatic index. So yeah, thank you for asking about this.
Okay, next question is also from Dr. Steve. Even though there was only one ionic bond making up about 2.1% of the total bonding, do you think this ionic bond is responsible for a lot more than 2.1% of the total interaction strength, since ionic bonds are normally stronger than van der Waals, for example?
I think there is a possibility, because yeah, ionic interaction is more stronger. But yeah, I think we need to look more on this, because previously there is a researchers showed that the interaction is mostly van der Waals and hydrogen, and this is the one that established the binding between CT and the receptor. So we can maybe look further for the ionic bonding, since it's a good suggestion. Yes.
Okay. Next question is, with how strongly the CT binds to your TNFR1, do you think that it's likely impossible to break this bond, for example, with a drug or antivenom, after the interaction has already occurred?
Wait, let me think. I'm not very sure about this one, but it is a very strong affinity between TNFR1 and CT, but it also can have a reversible reaction, as suggested by the intermolecular interaction. So maybe there is possibility to break this kind of interaction with the drug, antivenom, etc., as you suggested. So yeah.
Okay.
Maybe
Okay, thanks for the presentation. Any more questions?